gas is not so good for certain, for safety reasons. Nice. So thank you all. Anything else? Nice, 5.0. Yes. Nice, thank you. Very pure. Nice, thank you. And second one, what are the pros and cons of near-infrared spectroscopy to monitor emulsion polymerization reactors? You want to show answer? Maybe the other group now? Group 2, yes.
The pulse, the probe can be inserted into the reactor, eliminating the need of opening the reactor, and the analyses are fast. The pros, there is water interference, and then overlap, and the equipment requires calibration.
Nice, thank you. Anything else? Another group, maybe? Is it the same? No people, the same? Damasio, Mikael, and Maria Esther?
I also mentioned that the measurement needs to be very close to the surface. Actually, I don't know if Eder already told this, but it is due to the limited light penetration.
Yes. Yes, it can be inserted or very close to the surface, yes.
Mm-hmm.
Because we cannot measure in a fast way, we cannot measure because we do not have this penetration. It's not so big, this penetration way. Yes. The length of penetration. Nice. For sure. The good is that we do not need reactants, we do not need the gases, and we do not need the equipment in the industrial plant as chromatography. So we can have the probe and we can have the equipment in the other room, another place. It does not need to be closed. It's a very great advantage from this kind of technique, for sure. But we need calibration. That's not always good. But sometimes if you have once you have done it, it will work. So when we think about industrial production, you are always producing the same product or the same products. So you do a calibration for this kind of product, for this kind of process, and then you just do it. So it's not so bad needing calibration. It's an advantage, but it's not so bad. Yes, anything else? Mikhail, sorry. I was confused about the groups. Nice, the last one. Start with some batch combination experiments. Which monitoring technique is best suited to monitor free monomer? Calorimetry or Raman spectroscopy? And just the five. And then, which one? Which Which one have you choose?
If it contains only one type of polymer, the best technique is the colorimetric reaction. But if it has more than one type of monomer, Raman is more sustainable. Colorimetry is better suited if a single monomer type is the same as it non-invasive. So BIF is non-invasive, easy to implement, and provides a continuous response with no delay, with only one monomer. If there is two types of monomer being fit copolymerization, Raman will be more well suited for directed measurements. Able with a small delay and also more expensive process. We didn't know if have only one monomer or more than one monomer, so we put for the both.
Okay. Nice, thank you. Anything else? Anyone else? Yes.
The Raman spectrometer is the best suite because it has the advantage of doing a single measurement and directly measuring the amount of monomers unreacted and solids. In calorimetry, the general conversation is provided directly, but not the free amount of monomer, and is necessary continuous measurement.
Nice. Also nice. Anything else? Nothing. I'll just ask you, before I complement our answers, I'll just ask you one thing. What we measure, what property or what variable we measure in calorimetry, and which property or which variable we measure in Raman? Could you answer me, please? Can be in the chapter. Yes, calorimetry temperature, split flow, yes. And Raman, we measure temperature and we have heat flow, yes. Nice. And Raman, which variable do we have?
I don't know, but when we use Raman, we think it in the vibration of the molecules, but I don't know who is the what is the miserable. Yes, I think energy perhaps.
Yes, yeah, no, it's okay, it's okay. Yes, yes, you see the diffraction of light, yes, it's right. And what you are using is diffraction of light. This is the thing that you are measuring, the variable. Yes. And then we transform this to a spectra equipment, for sure, not a for a spectra in which we can associate these peaks and these bands to vibration of molecules. Yeah? And then we can identify the groups that we have on this substance. Nice. And here, what kind of vibration are we interested in measuring in the motion polymerization? The light refraction caused by each group. Double bond, yes? The double bond of the monomer. This is what we are directly measuring. The diffraction of light caused by the presence of this double bond of the monomer. And in calorimetry, we are directly measuring temperature and we associate this with heat flow. Okay, because we have the light is passing through the sample, that is the reaction medium, and we have some light diffraction that is caused by the substance that have in this medium. So in this case, we are interested in measuring how this light is diffracted by the double bond of the monomer, because we are interested in the presence of monomer, the amount of monomer that we have in this medium. So when we have the spectrum or the spectrum from this spectroscopy analysis, we will see the peaks, the bands associated of each group. And we are interested in seeing the signal of the peak associated to the double bond of the monomer, because the intensity of the signal is related to the concentration of this double bond, that is related to concentration of monomer in the medium. Is it clearer or not? Yes? So, nice. So then, what are we measuring? So, calorimetry, we are measuring temperature. And we know that temperature changes because of the reaction, because we have a polymerization reaction that is exothermic. So we are changing the temperature, we are changing the heat flow of this reaction. And Raman, we are measuring the diffraction of light caused by the presence and the amount of double bonds that you have. What means light diffraction caused by the amount of monomer. So see that Raman, we are measuring directly, almost directly, the amount of monomer. And calorimetry, we are measuring the heat flow and associated this to the amount of monomer that was converted to polymer. Okay, so your answers were all right, because we were just discussing the advantages and disadvantages and applications of both techniques. But when I think about a reaction, an experiment that's being carried out with a very low amount of monomer, that means starved semi-batch polymerization. You have a reactor, and then you are feeding the amount slowly. As you add feeding the monomer, slowly. As you add the monomer is consumed. You add more, it's consumed. You add more, it's consumed. What's happening here? You will not have a lot of monomer in the medium. So it's difficult to use, for example, Raman spectroscopy in this case. Why? Because Raman is measuring the amount of monomer. As it does not have a lot of monomer, it's difficult. This is the difficulty. But you are adding monomer, it's being reacted. You are adding monomer, it's being reacted. As it's being reacted, it's being consumed, and we have exothermic reaction. We have a heat lost here. the heat is being changing, a heat flow here. So you are measuring this heat associated to the reaction. Reaction is being continuously. Okay? So just to understand what we are measuring, it's important. So in this case, if you are working with a starved and batch polarization, what means that you do not have a lot of monomer, you have too low monomer, amount of monomer, Raman is not so good because we will not have monomer to see the spectra and to quantify the amount of monomer. It's better to use calorimetry because in calorimetry we are having a measurement of reaction rate, the heat from reaction rate. Yes? But of course, I agree with you. It depends also if we have only one monomer or two monomers. That's better. Is it clear or not? Same. Yes, for all of you. So it's very important to understand when we are using, and then we can apply this for all other areas. When we are using some analysis, some technique for analysis of some sample or some reaction, we need to understand the result that we want and what we are measuring to see if we are measuring this thing that we are measuring is suited for this technique. If the result is appropriate for this technique. Both things have to be considered. Yes, people or no? Do you agree with me? You can disagree if you want. I guess you're just thinking about. If you have any doubt, please feel free to say. Okay. Did you understand what is targets in Dutch conversation? It's a starved. It's the word starved. It's the same meaning as for food for humans. Yes, yes. Little amount and controlled amount. Yes, both words are right. So it's too low as I have it consumes before adding more. And that's why Raman is not so good because we will have almost, if we are looking to the reactor, the monomer that I add is consumed, is instantaneously consumed. So I will have very, very little amounts in the monomer in this reactor, not reacted monomer. Will not have this. The amount is will be too low. Okay? Yes, there will be no double bonds to measure. If you do not have monomer, you will have no double bonds to measure. So it will have no results for us, but reactions occurring. Because we're measuring the double bonds of monomer. As it is instantaneously consumed as it's added, we will not have these double bonds to measure. But reactions occurring, and then we have heat flows that we can measure. Nice. So do you have any other questions, doubts, or about something in the course? Next class we'll have the final test. That will be individual. If you have any doubt, this is the time for asking me. The right time. For sure, you can feel free if you study and if you have any other doubt, you are free to write me by email also. Please feel free. And in the other way, you'll have the test next week on Thursday, and but is there that will not be on Thursday? Yes? Is this?
Yes.
Okay.
My test will be Monday.
Yes, I already talked with Professor Claude about this. Nice, just to remember. And the other ones, it will be on Thursday morning, and will be presential, sure. So it will be a written test with some questions about our course, to be in English, but we accept answers in Portuguese if you prefer. For sure, we prefer in English, but if you are not so comfortable to write in English, you can write in Portuguese. Okay. So, if no one has any other thing to ask me, I would like to say that it was a pleasure to be your professor, and I will not be on Thursday, unfortunately. But it was a great pleasure to be with you in this course, with these classes, and to know you. Nice, thank you, ladies. And I expect that all of you have a great work in your master or PhD.
Thank you.
Okay. And then, Mazi, you have calculating kinetic measurements. If it will be in the test, is this your question?
You'll be question only for the part of the theory.
If we will have some calculation in the test, is this your doubt?
Yes.
Maybe we can have calculation, but not so. What can I say? Not so complex as we did for kinetic measurements. Okay. Maybe we can have some calculus in the test, but not so complex. We did in the computer with an Excel. It doesn't make sense to do it in the paper. Okay. Nice. So I'd like to thank you, and if you want to open your camera just to say goodbye, it would be good. If you do not want, it's okay for me also. And thank you, all of you. Thank you for your patience with these online classes. Besides, we are in presential, I did online classes. So thank you for understanding it. It's not so easy to go to Florianópolis only for the class. It's
Understand.
yes, I was tired of it, very tired, and it's also dangerous, not so good.
Yes.
So I just like to thank you for doing this with me, and thank you for this course, all of this course. Feel free if you have anything to share or to ask with me. Feel free to write to me. Okay, and those who will work with in the lab, in our lab, also feel free to contact me if you want something about polymerization, or non-encapsulation that I work. If you want to do the course of non-encapsulation techniques, it's also very interesting. And then you are all welcome. Okay?
Thank you.
Nice. So thank you. See you. And have a good test next week.
Okay.
Okay. I hope so. To be easy to correct.
Yeah.
I always hope that you do a good test because it's easier for me to correct. Nice people. It's good for you as well.
Okay.
Bye bye.
Bye, teacher.
Bye. See you.
Thank you.